Chapter 12, Sensory Mechanism, Screencast 4. Now, this screencast is focusing on the special sense of vision. So now we're going to be looking at visual stimuli. And what the receptors in the eye do are responsible for converting light energy to nerve impulses. And then those nerve impulses will be sent to the brain where they're going to be interpreted. Now, if we look at the structures, we want to kind of keep in mind what they are. So we have the sclera. That's actually the whites of the eye and cornea. The aqueous humor, that's going to be the iris, along with the iris and the lens. Then we have the vitreous humor, retina, optic nerve, macula, optic disc. One of the things that's going to be important is position in front of the lens versus behind the lens. So that's going to be one of the terms I use to split the anatomy of the eye itself. Now here you can actually see the overall view of the eye itself. And one of the things that I may ask you to do is to basically track the path of light through the structures. And I would go a linear direction. Okay, so that's something that you should be comfortable doing. So going from the path of light all the way through, okay, and ending with the optic nerve. So I want to make sure that you have that kind of idea. Now here, this could be how I work with a matching question with regards to the typical structure of the eye and the function. So once again, when I go to matching, I will typically go to these tables, and I think I indicated that on the review sheet. So this is something that I would ask in a matching type question. Now, if we look at a couple of features, so the first is the iris. And what the iris does, that's vascular, that's smooth muscle, and what it does, it regulates the amount of light. So the iris itself is what causes the pupil to constrict or dilate, and that actually controls the amount of light entering the eye. If you happen to be a photographer, I do a lot of photography, and one of the things that you do is basically an f-stop, and what that does, that's the size of the shutter and how much light is allowed to make its way to the sensor. So those of you that take pictures, that's something you might be familiar with. Now focusing, what that is, bending the light by the cornea and the lens. Now, accommodation, this is this process where adjustment of the lens allowing for focusing on near and far objects. And it's the ciliary muscles that are actually responsible for this. And one of the things that we do know that is and that aging impacts is how well we accommodate. And we know that vision typically deteriorates as we age. Now here we're looking at two different types of how the focus is set up. Now notice, once again, like optics, there's an inverted image, and that image, the brain actually flips it the way it's supposed to. So for near objects, what happens is the ciliary muscles reduce the tension on the fibers, and that allows the lens to bulge, and we see proper focus here. Okay, now here we're looking at focus on distant objects. So now those muscles are actually relaxed, and they kind of increases the tension on the fibers, and that pulls on the lens and flattens it. The goal here is you want things to be focused appropriately on the back of the eye, on the retina. Now, when we look at common disorders of vision, we have myopia, and that's nearsightedness. And the eye is actually longer than normal, okay? So what we see here, distant objects focus in front of the retina. We have hyperopia, and that's actually farsightedness. And so here, the eye is actually shorter than normal. So near objects focus behind the retina. So I'm not going to hold you responsible for the different types of lenses that are used to correct this, but you do want to be comfortable with them, okay? Now, with astigmatism, we're looking at an irregularity in the cornea or the lens. So some individuals that have an astigmatism, the lenses that they use in their glasses have to be ground specially that compensate for lens irregularities. Now, it's interesting because quite often, an astigmatism can go away. And in the past, I've had contact lenses specific for astigmatism, and then I go through a period of time where that astigmatism is no longer present, okay? But I tend to be actually very nearsighted, and that accounts for the misshape of the eye for the most part. And so we can actually see the overall issue with nearsightedness versus farsightedness. Okay, and the lenses that are used to actually correct this so that people can actually see normally. Now, the retina, that's where our photoreceptors are located. And the role of the retina, it allows us to see in color. We adapt to light intensities, and image perception is due to this particular structure. Now, there's multiple layers to the retina. So you would want to know these. Okay, the outermost, these are the pigmented cells, and they absorb excess light. The actual photoreceptors, those are the rods and the cones. And then we have bipolar cells, and they actually connect with the rods and cones. And the innermost layer are actually the ganglion cells, and the axons of those actually form the optic nerve. Now, here we can kind of look at the path of light and how it's actually moving through the layers of the retina. So we can see the various layers there, okay, as far as the outermost to innermost, okay. Now, what I want to point out here is what's actually called the optic disc, and that's a blind spot, okay. So it's right here overall where the optic nerve is leaving the eye. Okay, so you can actually see how well vascularized it is. That's important, okay? But now we see that blind spot. Now, the photoreceptors, and one of the things I want you to be comfortable with is what responds to what with regards to the rods and the cones. So these basically, they respond to light, and they're going to change their shape. Now, when we look at rods, just to get an idea, there's many more rods than there are cones. And generally, okay, we can see a fair amount, okay? There are lots of ganglion cells. And one of the things that's happening here is neural processing. So we see a lot of convergence and divergence and summation, convergence and summation occurring here in the generation of the action potential. So once again, we see convergence once again because the information is coming in. Convergence, not divergence, but definitely summation. Now here we can see the difference between a rod cell versus a cone cell. Okay, so there's definitely a structural difference between the two. Now, when we look at rods, okay, rods do not do color vision. That is something I will definitely ask. And there is a particular pigment, rhodopsin, within the rods, and that's actually we need vitamin A to actually produce rhodopsin. And you may have heard carrots are good for your vision, and so that's where we actually see vitamin A. Now, dim light vision is highly dependent upon the rods, and once again, rods do not enable color vision. And if we go back to the numbers once again, notice we have significantly more rods than cones. So rods work primarily in dim light. They do not enable color vision. And also there's not an equal distribution between rods and cones. So the regions of the retina farthest from the fovea, they have the highest ratio of rods to cones. Now, there's three types of cones that enable color vision. So red, green, and blue. You may have heard of RGB. That's another color scale that's used with technology quite often. And so it really stems from our ability for these colors. Now, the brain, what it does, it interprets the ratio of impulses coming from the ganglion cells that are connected to the three different kinds of cones. Now, cones, one thing that's important, they require much stronger light to be activated. So in dim light, we use our rods and we don't see color. Now, the other thing that cones are responsible for is what we refer to as visual acuity. So that's sharpness of vision. Now, when we look at things that how they adapt as far as our visual receptors, it's depending upon a couple of things. The adjustment of the pupil size by the iris, and also the adaptation by the rods. Now, adaptation takes longer when going from bright light to dim light. What happens basically, the bright light actually uses up the rhodopsin temporarily, and it takes time to resynthesize that. So that's quite often when we go from a very, very bright light, how it impacts our vision in low light situations. This is going to be a video that pretty much just concentrates on the function overall of the eye. So once again, I recommend that you take the time to review this. The last part of this screencast is focusing on general types of disorders of the ear and hearing. Now, deafness is overall defined as loss of hearing. And there's different types of hearing loss. And I think it's important because in the city of Rochester, we actually have a relatively high population of people that are deaf. Now, there's different types of hearing loss. You have deafness that's due to the actual damage to the hair cells. And then there's conduction deafness. And this is actually damage to the tympanic membrane or the bones of the middle ear. In some cases, this can actually be corrected with what's known as a cochlear implant. Now, another real common ear disorder is otitis media, and this is actually inflammation of the middle ear. And there's also a syndrome, Meniere's syndrome, and this is specific to the inner ear, and that impacts both hearing and balance. Now, if we look at some common disorders associated with vision, you may have heard of a retinal detachment, and this is actually common in those individuals that have those long, irregularly shaped eyeballs. There's extra stress, it kind of stretches the retina, and it increases its likelihood for detachment. Now, the retina actually pulls away from the choroid, and symptoms that are common with people that have retinal detachment are flashes of light, blurry vision, and also a loss of peripheral vision. Now, if this is detected early enough, it's something that can be surgically corrected. As we get older, some of us may actually develop cataracts, and that is when the lens actually becomes cloudy or opaque. And the treatment for this quite often is either a cataract transplant or replacing the lens with an artificial lens. Now, glaucoma, when you go to the eye doctor and they shoot that puff of air, really what they're doing is measuring the pressure of the inside of the eye. And what may happen is if you don't have the right draining of the aqueous humor, pressure builds up. And this can actually is treated with drugs or surgery. But if it's not treated, it may actually lead to blindness. Here we can basically see retinal detachment. Here we can actually see cataracts. Okay. Now, one of the more common that we see is age-related macular degeneration. And what happens here, the retina detaches and photoreceptors in the macula degenerate. And one of the things that we see here is the loss of visual acuity in the center of the visual field. So things just aren't as sharp. We see things a little bit more blurry. And when we look at the leading cause of blindness in people over the age of 60, it really is this macular degeneration. Now, some individuals may actually have what's known as color blindness, and with those individuals, they're lacking one of the three types of cones. And generally, these individuals have the inability to distinguish the full range of colors, and this will result in what's known as red-green color blindness, and this is often passed on or inherited. Here we can see normal vision. This is what we see with someone that has glaucoma, and here we see age-related macular degeneration. Many of you, when you've had your eyes checked, you've had probably, if you go to LensCrafter or any of those, they have a book and they actually ask you to identify these numbers that are hidden in colors. And so here, hopefully, you see number 12. And this is a general test used to identify color blindness.